Good morning, everyone. My name is Maya Luria with TMC for Seniors, and today we are going to be talking about genes, epigenetics, and the microbiome. Who is really in control? I have Jill Jones with me, along with Michelle Clark. How are you ladies today?
Good.
Doing well.
So you always do a wonderful job in sort of breaking down these sort of complicated topics and, doing it with some fun. There's always some great videos in there that really explains things well. So I'm so glad you're here with us today to talk about this. You do have an online audience that's watching along with an in-person audience. So if you are watching from home, please feel free to put any comments into the chat and I will address those afterwards. And I'm going to go ahead and put your PowerPoint up so you can get started. Thanks for being here.
All right. Thanks, Maya. Well, good morning, everybody.
Good morning.
Nice to see a full house this morning. She already introduced us, so you know who we are. I'm so used to introducing myself. Well, anyway, when Michelle and I decided on this topic, when we decide on any new topic, we start searching on what's the hot thing in health now, what's the hot topics, what's being talked about and in magazines and in newspapers and so forth. And over the last about 3 years or so, we keep getting the same answer, microbiome. And so Michelle and I have been doing classes on the gut microbiome for over 3 years. And we dug a little deeper to see where the research is being done and where the money is being spent. And it turns out another big influence on our health is our epigenome, our epigenetic markers. Are you familiar with that? Anybody familiar? You are? Okay, good. But so what we thought we would do for today's presentation is look at all three systems, our DNA that we inherit, our epigenetic marks that we acquire during our lifetime, and our friendly bacteria and microbes that live in our gut. So we're going to look at all three systems and talk a little bit about each one, describe each one, and then show how they work together. And then the last part is always how can we influence a healthy epigenome and microbiome. Okay, so here are, as usual, our research, our information comes from the top academic journals like the Cell collection of journals and Nature. And I have one down there in the corner that's a 30-year-old Scientific American. And I'm going to talk a little bit about that later. So here's the outline for today. If we want to understand our body, we need to talk our body's language. And the language of the body is chemistry. Molecules are how the body communicates, how the different cells communicate and stuff. So it's about chemistry, and I'll do a little molecule 101 in the beginning just to set it up. Then I'll talk about our genes, what we inherit, and what they are, what they do, and the epigenome. And I know this is new to a lot of people. It's the markers, they're chemical markers, molecules, that get laid down on top of the DNA that affect how genes are expressed. And I'll get more into that. And then, of course, the gut microbiome, we'll talk a little bit about that. And then how do the compounds and chemicals being created by the bugs in our gut, how does that affect our genes and our gene expression? And then connect all of that to food. How does that influence good food choices for us? And then some guiding principles, not rules or diets, but guiding principles for food that gives us overall good health. When we look at a plate of food and we're describing it to somebody, we're usually saying, oh, the salmon was done just right, the rice tastes a little bit nutty, or the mushrooms are great, they had a little bit woody flavor. But when our body sees a plate of food, it it thinks in molecules. It sees a bunch of molecules that are going to be delivered to different cells in our body. And so, for instance, when we're talking molecules, water, we know what water is, H2O, an oxygen and 2 hydrogens. So that's called a ball and stick model. And that's how molecules are represented. There'll be a ball and stick model, chemical formula, and there's other representations. We're going to be using the ball and stick, like carbon dioxide. There's the atoms that are in it and the bonds. Those sticks are the bond, the chemical bonds. And then if you put 2 of them together, they get transformed into a whole new molecule. This one happens to be seltzer water or fizzy water. It's carbonated water if you put carbon with oxygen. So anyway, all this molecules, atoms and molecules coming together and interacting, that's chemistry. And so I view it this way. Chemistry is kind of like the language of not just our body, but of life in general. Everything is about molecules and signals. So a plate of food really gets broken down into molecules, and molecules are the building blocks of our cells. And of course, our cells are the foundation for life. And that's biology. So we're moving from chemistry to biology, and in the middle there, we get biochemistry when we're talking about chemistry inside our body. But so the question here for you is, what do you think is the most important molecule of life? Well, frame it as a Jeopardy question. I'll show you.
There he is, our Alex.
Actually, you were the first one. It's DNA. DNA is a very big molecule, very complex molecule. But it was Watson and Crick who discovered the structure of this molecule, the 3-dimensional structure of DNA. And this is a ball and stick model. It has all the atoms and how they're bonded together. Before Watson and Crick came up with this, the ingredients were known, the atoms were known, and chromosomes were known, but they didn't know how it was structured. And this structure makes all the difference, because now they understand how DNA can actually copy itself. And DNA, when you hear about it in a cell, if you stretched it all out, it's about 6 feet long. But it's not just one 6-foot molecule. The molecules are broken up into chromosomes. Sorry, my mouth is really dry. So the molecules, and you've all heard this, we have 23 from our mom and 23 from our dad, 46 molecules. But these are all molecules of DNA. The question is, why do you think it's the most important molecule in biology? Why is it the most important molecule of life? And the reason is it's so unique. It stores all this information, all the information about how this person is going to be built and maintained, all the function and features of a human being. That information is stored and it's preserved. When the cells divide, that same exact DNA gets copied from cell to cell and from generation to generation. That's what makes it so unique. It's like a whole memory system in here on how to build a human, in our case humans, but other life forms. So if you took one of those chromosomes and kind of pulled it on the end and stretched it out, now you can see the parts of DNA. And the backbone, we call it a twisted ladder. So the sides of the ladder are sugar and phosphates and acid. So we have bases, the rungs are different bases in the middle here. And there's 4 different ones, and they show up in pairs. So the rungs there, the A, T, C, G, those are the names of them are adenine, guanine, and so forth, but people only use the letters because that's easier when they're looking at the whole or sequencing your genes or looking at the genetic code. That's the genetic code, all of those letters. And there's 4 different ones, and an A always pairs with a T and a C always pairs with a G. It's just how it works. Now, so what are genes? Well, if you take all of this DNA and look at one segment of it, and you look at the base pairs, a gene is a set of those base pairs. It's a segment of DNA. And now I'm just showing a few here, but a gene may be 10,000 of these base pairs. Because in our whole DNA molecule, we've got about 6 billion base pairs. And so roughly 10,000 of them make up a gene. And a gene doesn't really do anything. It's a set of instructions telling your cell what to do. So a single gene is a set of instructions, and it's giving your cell orders on what it should do, how it's going to build a protein. So normally you have one gene giving the instructions for building a specific protein. And just lately now they're saying that it can be instructions for more than different variations of a protein. So anyway, a gene, their whole job is to provide instructions to build a protein. So what are proteins? We hear this term a lot, but almost everything in our body is protein. Our hormones, enzymes, all of our cells, all of our muscles and skin and everything is proteins. And so the way our body views this, or the way the cell views it, it gets instructions from the gene to collect a group of amino acids. And anybody know how many amino acids are out there? There's 20. And mostly what we'll hear about is 9 essential. And essential amino acids, those are the ones you have to get from food. That's why they're called essential. Your body cannot make those. Body can make everything else, but for 9 of them, we have to get it from food. So the question here is, a lot of people are saying, do I get enough protein? How much protein should I have? The other part of that, though, is, am I getting all of those amino acids I need? So anyway, the gene gives instructions to the cell. Cell gathers up these amino acids and puts them together in a chain. And so what's important is that it gets the sequence right. Every one of these amino acids has to be in the right sequence to build this protein. And then after the chain is built, got all the amino acids there now, it starts getting twisted and transformed and moved into a certain shape. And that shape is really important because the shape is what determines the function of the protein. So we want to have all the right amino acids lined up and then put into the right shape. And I'm going to show you an example of insulin happens to be a protein. So if we look at insulin, and its job is to regulate blood sugar, insulin has 51 amino acids, which means a lot of them are repeated. So, but it also has 2 chains. It's chain A and B. When you have 2 chains like that of amino acids, that designates a protein. But if you only have 1 chain, that's peptide. So you hear that term peptide a lot. All it means is it's made up of amino acids. It's just one long string. So once they're all together in their right sequence, then they all get folded up and get folded into the right shape to do the job. Does that make sense? Mm-hmm. Okay. Well, what do proteins do in our body? They do a lot of different things. But we talked about hormones. They move things around the body, like red blood cells carry oxygen. A big thing is enzymes. We have so many enzymes because at any point in time, we've got 1 trillion chemical reactions going on in our body, and it's enzymes that are the catalyst for that. Antibodies are made of proteins and all of our, of course, our body structure. So this is kind of a summary, and it's pretty simplistic. A gene is a set of instructions to build a protein. When a gene is doing this, when this is happening, that's called gene expression. When a gene is turned on and it's telling the cell to build certain proteins, that's what gene expression is. If the gene is turned off, then if the gene is turned off, it's not being expressed. If it's turned on, then it'll do its thing. And here's just a simple slide that shows how gene expression is defined. Our gene is a set of instructions, tells the cell how to put the amino acids together, and when the protein's built, that will influence our traits. For instance, eye color, it's not just one gene. There are a lot of genes that influence the eye color trait. But a lot of our different features of our body have these different traits. And so, are there any questions on this? Okay, what I do have here, because it's hard to explain in a few minutes, I have a little movie that pulls it all together. So if I forgot anything or didn't explain anything well, we'll show this movie.
DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other, kind of like Legos, to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind, there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which you don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine, AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life.
I didn't want to try to explain RNA. and that's not really the important part, but one thing I did want you to pick up from this movie is that our DNA stays protected inside the nucleus of a cell. So it doesn't leave the nucleus, just a short little copy of it leaves the nucleus and goes out into the cell and then builds the protein. Here's a quick look at proteins again. I think I've probably explained that enough. It's just, it's a way of packaging the DNA, and especially packaging it that way whenever it gets copied. It makes it easier to copy when they're all together organized in these chromosomes. The other thing to know about chromosomes is that they're only visible like this at a certain stage of cell division, when a cell's dividing, because like the movie said, they're normally all mixed around like a bunch of noodles inside the nucleus. But then they start to get organized whenever they're going to be copied and make a new cell. This happens to be, the other one was a textbook picture. This happens to be a real person. And I don't know if you've had any, where you've seen any of your chromosomes on a chart or anything, but each panel here are the chromosomes in different cells. So that's 3 different cells there. Karyotype just says it's a picture of your chromosomes. But the important thing is that every cell in our body has the exact, all our chromosomes, all the genes, all the DNA. The DNA is the same in every single cell. Well, what makes the difference between a heart cell and a liver cell or an eye cell? What happens is some genes are turned on and some genes are turned off. So in your heart tissue, you won't have liver genes turned on, you'll have them turned off. So the different cells know what to do based on the genes that are turned on and how they're expressed. But anyway, when you look, this is a real person, as I said, and these were all normal, a little bit beat up by the time you're in your 70s, but they still look good, they're normal. And what the doctor is looking for is to see if there are any anomalies or any problems with any of these chromosomes. Something can be missing, something can be organized wrong. Those bands across there, that's a stain. So this particular cell had a stain put in it at this stage of reproduction. And the chromosomes pick up certain colors. They'll pick up a light color or a dark color. The light bands, what those mean is that those are the genes that are being expressed. It's a group of genes in this chromosome that are being expressed in the light areas. In the dark areas, those genes are turned off. So if you ever get a copy of your own chromosomes, you can look at that and know. There'll be other details on there too about the individual cells and individual genes. So we're going to go back to now our little piece of DNA. When you look at all those 6 billion base pairs, only 2% of them are actual genes. So only 2% of all our DNA is genes. The rest of it is called non-coding DNA. And they used to call it junk DNA. So it doesn't do anything. It's only the genes that are important. But as you probably are well aware nowadays, they know that these non-coding DNA segments here, they have a function. They help, again, guide the genes that are turned on and help guide that gene expression. So it's kind of like dials and switches for our genes. And we even have some of these, like I said, a gene could be 10,000 base pairs. In between there will be some non-coding areas. When you hear the term genome, the definition of a genome is the entire set of DNA in the cell. It includes the non-coding and the coding part, all 6 billion base pairs. That's a genome. That's our human genome. That's what we inherit, and that's what gets copied from cell to cell and passed on to future generations. So that doesn't change. When you have identical twins, they get an identical copy of that whole DNA, all those chromosomes. They have identical copies. All those base pairs, coding and non-coding, they're all the same. But then as they get older, like here's a lady, she's a twin. This is her twin sister. And if you put them side by side, they don't look the same anymore. And the one on the right has more health problems. And it turns out the one on the right was a heavy smoker. And she spent a lot of time in the sun, so she had those two. Those were the main factors causing her health issues and her difference.
One of the most important areas in biology at that point was going to be the study of epigenetic regulation of gene expression.
Epigenetics plays a crucial role in regulating biological processes by chemically modifying our DNA with epigenetics in our genes. Every single cell in the human body has largely identical DNA. The DNA in each of these cells is 2 meters long. To fit into the tiny space inside each cell, DNA is packed down into a microscopic size using packaging material. Through a process called DNA methylation, little chemical tags called methyl molecules attach to this packaging material to turn the genes on and off. If the gene is turned on, it's active and can be read. If it's turned off, it cannot be utilized.
I like to think of the definition of epigenetics in a very simple way, and that I think of the genome as being comparable to the hardware of a computer. And the epigenome, which is above the genome, above the computer, is the software that tells that computer when, where, and how to work. So this is why you have the exact same genome in every cell in your body, but yet you have 260 different cell types in your body doing completely different jobs.
I kind of forgot that movie was coming up, so I didn't introduce it. I should have done a better job of that. But anyway, this is starting now to discuss epigenetics. As if things weren't complicated enough with all the DNA, now we add another layer on top of that called epigenetics. And as he described them, epigenetics are, we have our DNA, all those base pairs, but along with that, we have these tags, these chemical tags that are, there are about 3 different kinds, but people mostly talk about this one type of chemical tag because it's the most studied, it's the easiest to measure and so forth. But all of us have on top of our DNA, we have these chemical tags that tell the gene, again, they either turn the gene on or off. So along with the non-coding DNA, you have these two levels of systems that are giving instructions to the genes or making sure they're expressed in a certain way. So epigenetics, the study of epigenetics, is all these chemical tags that are sitting on this whole string of DNA that are turning these genes on and off. And we acquire them over our life. We have some whenever we're born to get started, and then as we age, these epigenetic tags will start to change. Aging, our lifestyle affects them big time, and that's where we have some control as well. The longevity doctors and anti-aging doctors and all of that, they can look at our DNA and look at the epigenetic tags, and they look for certain patterns, because different patterns for these marks, that tells how our body is aging. It will tell people how our body is aging. Now, the good thing about these tags, like I said, our lifestyle does influence on how well we sleep, how well we're eating, whether we exercise or not. That's going to affect these epigenetic patterns. But the good thing is some of them can be reversed or changed. If you have epigenetic marks that are increasing your risk factors for heart disease or high blood pressure or whatever it is, you can also, by changing your lifestyle, reset some of those markers and turn back the clock a little bit. It takes time though. These don't change overnight, but you can reset some of them with a healthy lifestyle, whatever age you are. Now, the question is, does the new baby inherit the epigenetic patterns of its parents? What do you think? Well, what happens is when the sperm is moving towards its target, the egg, the sperm and the egg do have the mother and dad's whole lifespan of epigenetic markers in there. But when fertilization takes place, it's like a miracle happens and all those tags get erased. Mm-hmm. And that new baby, that little fetus or the embryo, is starting to build a whole brand new set of these epigenetic markers. Does that make sense? Mm-hmm. Now, what happens is, so in other words, it's starting out in the womb as a cell starts dividing and making the organs of the baby and everything. These epigenetic markers are being laid down on top of its DNA. And it's a really dynamic period because they're being laid down and erased and put on and then erased. And that just goes on and on. But this whole baby now is getting its epigenome shaped, and it's going to be setting it up for life. It turns out, in some cases, there are exceptions, it seems like, to everything. But some of these tags, or some of these epigenetic markers, do not get erased from the parent. Somehow they get imprinted and the baby does pick it up. But for most of it, it's totally erased. It's starting out with a brand new set of epigenetic tags. The other thing that's important here is that the mother's placenta, whatever the mother's behavior is going to affect this epigenetic programming. So if the mother's a smoker or is under a lot of stress or something, that will be reflected in the baby's epigenome. This is a critical period when the baby's born up to about the age of 5. Again, everything's so malleable, the brain's malleable, so the epigenetic markers in the brain's DNA, all of that is changing and being established. And whether the baby during this time period is getting enough attention, are there any traumas in the young child's life, that could set those patterns in place for a lifetime. But all of this going on in that critical period, like I said, about 0 to 5 years old, is shaping long-term health. So our health was shaped very early on, and then as we grew and aged along the way, we've been changing these epigenetic markers and these patterns. Now, if we want to, and we can still, even at our age today, we can make improvements. The number one powerful way to influence these marks is through the food we eat, because food is those molecules, it's the building blocks of our body.
Is we're not just running on food, we're also manufacturing ourselves out of food. That's particularly vivid when you think about a growing child. You see a child 1 year and you see that same child the next year and they've grown this much. Where did that extra Jill or Johnny come from? Well, it came from food and nowhere else. We literally are what we eat. It's not a direct translation. You don't eat French fries and turn into French fries, but you extract the nutrients from what you eat. Those become raw materials in the construction project of building the growing body of a child. And that growth comes directly from food. In the case of an adult, we're reconstituting ourselves on a regular basis, turning over hundreds of millions of cells, replacing spent hormones and enzymes. So food is both our fuel and our construction material.
Well, after he said that, I thought that was a good introduction of the importance of food and how it's these building materials. I asked one of the AI tools to come up with, to show a picture of what we would look like if we looked like the food we ate. And it came up with a healthy version.
Where's the chocolate?
I know, where's the chocolate? But I thought, that's kind of the thought that entered my mind. We're built for food, and you think, gosh, what would I look like now? So anyway, if you look at your body as a construction project here, the genes would be the blueprint of how things are supposed to look, and the food are the raw materials. So we want to have quality materials just like we would if we were building a house. And then the body, it's not just one and done. We're constantly refreshing our cells, where old cells are dying off, new cells are being reproduced, and all of that maintenance and repair is dependent on the food we eat. Major dependent. Now, to understand how our body takes food, turns it into molecules, absorbs it into the body, and has it go to the right places, we want to talk a little bit about how that happens. And it starts out, once you've eaten the food, it gets into the gut and it has to be broken down. Michelle and I, like I said, we've been doing presentations on the gut microbiome for 3 years, and all we were talking about is the colon, almost like the colon is the whole story. But that's not true. Most of the food we eat is absorbed through the small intestine and gets into the bloodstream. All the simple carbs get absorbed in the small intestine, proteins, fats, all the vitamins and minerals, that gets absorbed before the colon does anything. And why this matters is because now those are the building materials. Those are the building materials that are going to make all those enzymes and hormones and cells and tissue. So I have a little animation to show how that works. You eat a plate of food, protein gets broken down into those amino acids in the small intestine. Starch gets broken down into glucose, fat gets broken down into fatty acids. So between the stomach breaking things down and the small intestine breaking things down, now it can get absorbed into, has a few other phases here, but it gets absorbed and enters the bloodstream. Absorbed means it passes through the small intestine into our body, and then it makes its way into the bloodstream. And that's where it can affect the DNA in our cells, that epigenetic pattern in our cells. What happens in the colon, any of the food that did not get absorbed up in the, or broken down or absorbed up in the small intestine, that gets handled by the microbes in our gut. The microbes will break down the fiber and any other big molecules that could not get processed in the small intestine. Those big molecules wind up down in the colon, and that's where the microbes take over. So fiber-rich foods like vegetables that have a lot of fiber and fermented foods, and also phytochemicals. Those are the chemicals that plants make. And we've heard of polyphenols and all that. Those are all under an umbrella term, phytochemicals that plants make. They are important for our health as well. And that's kind of the big area where a lot of research is going on to figure out all these different phytochemicals, what they are, what they do, because plants make thousands of them. And they are beneficial for us. They're antioxidants. But anyway, they're a very big molecule. And so we have to depend on our gut bacteria to process these phytochemicals in order to get the benefit. So what happens is bacteria are fermenting fiber, and those byproducts send signals around the system. And those signals now will enter the bloodstream, and then they influence the epigenetic patterns. And they can even reverse. If we start moving to a healthy diet, they can even improve the epigenetic pattern. But again, over time, we're talking months or even years. So here are the principles or the guidelines for choosing healthy foods. Number one, there is eat a variety of vegetables, especially vegetables, fruits, and plants. And plants include coffee, tea. It includes olive oil, and all those things are considered plant-based. And also, feed your bacteria, the kind of foods they like. They like phytochemicals, and they like high fiber. And I'm not going in great detail with all of this because I've talked about it so much before, and I'm sure you've read about it. I will break them down, though. For instance, protein. Some people choose to be vegans. They just have to be really conscious of the amino acids. They have to make sure they're mixing the right proteins or plants together in order to get those 9 essential amino acids. Like rice and beans, for instance, would give you the full essential amino acids. But if you choose to eat meat or beef or anything, just try to get a variety of everything because they're all giving you different types of protein. And then again, here's the phytochemicals. They are anti-inflammatory and antioxidant. It's like having an insurance policy. So we're not going to be eating perfectly all the time. We might be eating a lot of chocolate or a lot of sweets or something. That's going to create waste, free radicals, they call it. That's where the antioxidants come in to clean up all that waste material. So antioxidants are kind of like an insurance policy. What phytochemicals do, the reason plants make these phytochemicals is because phytochemicals defend plants against pests, against bugs that might eat them or against diseases, but they're like the plant's immune system. That's why they make so many different kinds of chemicals. They also make them to attract pollinators. So they make certain chemicals that then are going to send off a signal to bees and whatever other pollinators to come and pollinate. And then they also use these they also make phytochemicals to communicate with the bacteria in the soil. So I think you might have attended one of our talks where we talked about the bacteria in the soil and how it communicates with the plants and the plant roots. Those are all signals, and that's where that comes in. As for our part, what they do for us, they are, like I said, they're antioxidants and anti-inflammatory. And some of these phytochemicals really help boost our energy. They go straight to our cells and affect, if you remember the term mitochondria, the energy producers in our cells. Now, so what protects the plants also protects us. I'm bringing back this little Scientific American, September of 1996, 30 years ago. I happened to be in Burbank Airport and was looking for something to read, and on the magazine rack was this special issue of Scientific American all about cancer. I was in my 40s, and I felt fine, never sick or anything, but it actually had a section in there that are warning signs about different kinds of cancer. And it turned out, I realized I had some warning signs for colon cancer in my 40s. Long story short, I went to a doctor and found out I did have colon cancer. And I was able to correct it at the time, though. But it was kind of amazing. But the interesting thing in that magazine, which I always remembered, was this quote: "The links between diet and cancer, however, may have as much to do with what is not in a diet as with what is. Skimping on vegetables and fruits can be a significant contributor to many different kinds of cancer. For reasons that are not fully known." They didn't know why. They didn't know all about these phytochemicals and polyphenols, and they didn't know everything we know today. Now we know those chemicals are anti-inflammatory and antioxidants. But I thought that was really interesting that even back 30 years ago, we were told to eat our fruits and vegetables. I just want to take a few minutes and go over the fermentation process. Fermentation, here's the definition of it. Microbes break down the sugars in the different plants or vegetables. If you have a jar here, microbes break it down and create new compounds, new textures, new flavors. But mostly it's the byproducts that it's creating that are good for our health. And when you look at this jar, that's the same thing that's happening in our colon. With the bacteria and the microbes that are in our colon, whenever we give them fiber or give them any of these phytochemicals, our colon is a fermentation tank. It's fermenting all of these plants and making the compounds and the byproducts in our colon, and then that'll get delivered to our cells. Now, you've heard certain berries and different plants and vegetables are called superfoods. Well, I think fermented foods are magic. And there's a lot of new research in this area, new findings. They're unique because of these live bacteria that we can have in our body temporarily. When we consume these foods with live bacteria, they don't stay there and take up residence. What the new research is showing is that they're not designed to live in our colons, but they're there long enough to produce all these great byproducts and chemicals. And when you eat fermented food, when you eat fermented food, you'll be getting live bacteria, but also dead ones. And even the dead ones benefit our immune system and our overall health. And the way it works is, again, these are all the benefits which you might know if you've read anything about the microbiome. but they protect our gut barrier, they help our immune system, they improve our immune system. And they can alter epigenetic marks.
So what's the fermentation process?
The fermentation process is in a jar, you've got salt water.
Yeah.
So there's a certain amount of salt that you put in the water, and that's a catalyst now to get the microbes activated. There's microbes already in the plants that you cut up and put in the jar. They'll get active and they'll start reproducing and start making more and more microbes, but they'll also be making chemical compounds and byproducts.
Yeah.
But it's the salt water that draws that out. You can do this at home. You can ferment vegetables and stuff at home. I don't, because you have to have the exact right amount of salt. You have to weigh it. You have to weigh your vegetables, weigh the water. The proportions have to be just right, or you could be growing something you don't want to. Yeah.
And let them know too that yogurt's fermented and kombucha and kefir, those are also fermented.
Yeah, there's a whole bunch of
So you can, if you can't get the vegetables, you can go to the yogurt, the kefir.
And this is just a cartoon to show that the dead bugs are just as good as the alive bugs. And again, this is really big news because we were told that our probiotic pills and everything didn't work, go to fermented food, well, all that's changed now. If the probiotic that you're taking, like if it's in a capsule or something, if it really has the bugs in it like it says, even if they're dead, they're doing good work. So I started taking probiotics again. We're getting close to the end here, but healthy fats are important. And what's healthy fats? Omega-3 is very important for the brain because the brain, as it sits here, the live brain is 70% water. But if you look at the 30% that's material, that's dry matter, that's 70%. 60% of that is fat. 60% of our brain, and especially in the area, the frontal cortex here, where we have a lot of our cognitive abilities, that's where the omega-3 fat is used very heavily. And when you're looking at omega-3 fats, well, there's different types of omega-3s. So if you choose to take a supplement in order to get enough of these omega-3s, what you want to look for is EPA and DHA. There's the certain types of omega-3s that our brain really needs, and the rest of our body, but our brain in particular.
And that's it. That's what we need, is that label.
I'm just showing you this label, and it's in your notes. You can choose whatever formula you want. I'm just showing you that it's a EPA and a DHA that you want to look for. And most of us, we want to add the two of those together. It should come out to roughly 1,000 if you're looking for a supplement. Now you can, omega-3s are in salmon and oily fish, salmon and sardines and whatever other oily fish there are. But I eat a lot of salmon, so I take one of these once in a while. But since I eat salmon probably 4 or 5 times a month, I think I'll probably have enough.
And
Okay.
that's in our handout.
Yes, all of that is in your handout. And I'm not recommending this as the brand you should get.
I know.
I just want to show you the label and what to look for in the different labels. And here we talk about reducing the amount of ultra-processed food. Well, now they're finding out that some of the ultra-processed foods are okay. It's really just this type, the kind of the junk food type, that are really bad for us. But there are some foods that are processed or ultra-processed that aren't really doing any harm. So you want to look at the ones like this that have a lot of sugar and fat in them as well. And of course, the last question if you wanted to pick a diet, the best diet, it keeps showing up over and over again in all of these longevity studies and well, I can't even say that, I won't go there, I can't say that word. But all these long-term studies where they've been studying people across their lifespan, the Mediterranean style of eating seems to be the one that wins to benefit good health in every way, our epigenetic patterns, our microbes, our overall health. And the reason the Mediterranean style is popular is because it's so flexible. It meets all of those guidelines that are the core principles for overall health. So I just have one last slide to end with. Now you know where my time goes. Okay, we'll get to the questions here in a minute.
So, yeah, you'll get back there, you can find a couple.
Okay.
Yeah, thank you so much, Jill. I do have a couple of questions that came in. Can you share sort of the difference or significance of probiotics and prebiotics, and do you need both?
Oh, yeah, we do need both. Prebiotics are what prebiotics are what the bacteria eat. So prebiotics are asparagus and broccoli and all the different types of plants that they eat. And now they're making these prebiotic powders. And now that's up to you. I can't really recommend it one way or another. Hopefully you wouldn't need, it's always better to eat the food if you can get it from that. Probiotics are live bacteria.
Okay. Now, what about, where can we go to get a fermenting recipe?
Well, online is one place. There are books. You can, Google books on Amazon and look for how to ferment vegetables or how to do fermentation. There are a ton of online videos that show you how to do fermentation, and you could just Google that on YouTube, which is what I did. And I bought all the equipment and was going to try it, but then I got scared. I didn't want to grow anything unusual.
Okay. Somebody asked about what ratio of EPA and DHA, I guess for omega-3s, is the best?
I don't know that there's anyone that's best. It's just as long as you get both of them. I'm not aware of any guideline that says this is the best combination.
Okay.
Because usually when you get them in a supplement, they'll be pretty close.
Okay. Another question was regarding salmon. I know you had mentioned that you like to eat a lot of salmon. Is there any issues with the mercury accumulation from eating that? Do you know?
Any issue with what?
Mercury?
Oh, mercury. Salmon is lower in mercury than most fish. So you can even look at a chart online and see what fish has the most mercury, but salmon is very low.
Okay. Do probiotics make it past the stomach acid to do any good? Was another question that just came in.
Well, everybody's an individual, and a lot of the probiotics won't make it past the stomach acid. They will get killed. But now the research is showing it doesn't matter because those parts of that dead bacteria are still doing good for our immune system. And they're calling it like a vaccine effect. It's because these dead bacteria are training your immune system that they're okay.
Oh, okay.
So showing your immune system that these are okay, you don't have to overreact.
Sure. Somebody also mentioned that they purchased a fermenting kit, and it included recipes, jar valves, and everything to put into the food jar, just sort of to stick to compact the food into the jar. So, so just for folks that were wondering about that as well. So
Yeah, you can buy kits. I didn't bother.
yeah, no worries. I want to thank you so much for being here today, Jill. So thank you for being here. I'm going to wrap up with our online audience and I will send it back over to you. So thanks again for being here. Thank you again for joining us, and we look forward to having you back. Our May calendar is out, and our next presentation is From Shoebox to Shared Treasures, A Beginner's Guide to Digital Preservation. That'll be on Tuesday, May 5th at 10 a.m. With the University of Arizona Libraries. It is called the Wildcats Digital Labs. That will be coming. So it's sort of a fun thing to learn how that you can preserve your digital memories. If you would like to sign up for this class, please feel free to give us a call. Our phone number is 520-324-1960. We'd be happy to get you signed up. That will be again on Tuesday, May 5th at 10 a.m. Then the next one is going to be on May 6th. That is Mindfulness Tools for Healthy Aging. And that is with Greg Dobler with our TMC Hospice. And that is, again, May 6th at 10 a.m. And we still have some spaces available for that class as well. So, again, the phone number is 520-324-1960. Have a great day. Thank you.